Good morning, guys. I know I'm a few minutes early. I just wanted to open in case I needed to upload my Zoom, update my Zoom. So we'll wait a few minutes. Good morning, guys. If you have any questions about anything right now, I'm happy to address those now too. But otherwise, we'll wait a few minutes. Okay, people are making their way into the room. That's always good. Make sure that this is good morning, everyone. Make sure that's plugged in. Happy last week of classes, right? Isn't it? Survived another virtual semester. Okay. I guess we'll go ahead and get started with some of the kind of housekeeping things we have to keep in mind. And then we will have, I'm very excited about today because today is our long anticipated first out of 2 seminars featuring special guests. So I've been very much looking forward to today's class. So hopefully we're able to get through it without a lot of technical difficulties, but I think we'll be okay. In the meantime, whoa, all right, someone needs to mute. Who there? Ahmed, mute. There we go. All right. Cool. All right, let me pull up these this little thing here. Wait, where did it go? Hold on. Share screen, come on, Zoom, share. All right. Hopefully you guys can see the PowerPoint slides. Let me fix this. All righty, and let me just look to see here. All right, we'll get started, and we'll hopefully our special guest will be arriving shortly. Yeah, so this is the second to last Planet Kingdom class for the semester, and today we're doing something different, which is we're gonna have these special guests come speak to us to tell us about their experiences working with plants and maybe a bit about their journey getting there and basically whatever they would like to tell us. They have the reins. Yes, so just a few reminders before we kind of get into that is we have a phonology assignment due on the 29th. Ooh, Claire is here. Hi, Claire. Due the 29th, right? Some of you have sent me the phonology assignment already. Some of you have emailed me about having issues uploading a portion or maybe both portions of your submission. And I'll get to those emails later today. But keep in mind, yeah, your phonology assignment is due the 29th, which is what, Friday? I think it's Friday. You also have your lab exam. Oh, sorry, no, the 29th is Thursday. That's right, Thursday. That's our last day of classes. Friday, you have your second lab exam. Yep, looking on the cal, that's correct. Last night, we had a extra credit assignment at the Torrey Botanical Society at the New York Botanical Garden. I was really happy to see a lot of you there. Thank you so much. I know it meant a lot to the speaker to have such a big audience. So that was really, really great. And thanks, Rose, for having the courage to ask a question. I was happy to see that as well. And then our final exam lecture is on the 11th, right? Which seems very far away, but it's not really that far away. And then I've also sent out to you guys a link, or it's also included here, to take a survey for this course to tell me what you liked, what you didn't like, what aspects you'd like me to improve, maybe things you'd like me to add. Like I said, this is the 4th time that I've lectured this course, and I've always used student feedback to really guide how I manage this course. So your feedback is super important to me, and it is completely anonymous. I won't even see the results of the survey until much later this summer, and I can't tell who is who. So please feel free to just be honest. Yeah. All right, so we're 10:05. Today is the first of 2 special guest lecture series that we're doing here in Plant Kingdom today. And today's guests, I love all the guests that we're having are amazing and represent different parts of the field of working with plants. And so I'm very excited today to welcome Dr. Georgia Silvera Siemens, Stevens Nemi, and Claire Hopkins. So up first is Dr. Georgia Siemens, but I'm not sure if she's here yet. Let me just take a peek. We're running a little bit ahead of schedule, as it were. So I guess I can back up while we wait a few minutes. Does anyone, have you guys, I will be posting a study guide for the last lecture exam on Friday. But does anyone have any questions about anything, or has anyone had issues kind of uploading the phonology assignments and wants to get feedback about that, or just any questions in general while we wait a few minutes?
Professor?
Yeah, how's it going?
Okay, for the Excel sheet, it has to be in PDF form, right?
No, the Excel submission can just be an Excel submission. Doesn't have to be converted to a PDF.
I already converted it, so is that okay, or do I have to go back and switch it?
Don't go back and switch it, but do send me a copy of the Excel file in an email, just so I have it. So part of the reason I do these phonology assignments is I've been doing them for 4 years now in this class every time I've lectured it, and I have hopes of maybe one day using the data that you guys gather over the course of that 6-week observation period as part of a kind of snapshot. I can't speak. Snapshot of phonology changes in phonology over time in our region. So that's the idea, at least. Depending on what the photos that you guys attach look like, it may be difficult for me to determine what species we're talking about. So there's that. But yeah, so it'll be useful to me to have the Excel file as an Excel file in case I want to grab, copy and paste data from those cells into a master file.
Okay, thank you.
Yeah, no problem. Any other questions, comments, concerns? Ben says, I wasn't able to attend a lecture last night. Was it good? I thought it was good. I thought it was really, really good. I was a little surprised how gene heavy it was. It was very, Miriam says she learned a lot. So it was a little heavier in the genetics than I was anticipating. So I hope you guys didn't feel overwhelmed from that. But I was getting excited when, especially during the introduction section where it was mentioned about heterospory and things like that. And I was like, my students know what that is. We've covered that. And yeah, Miriam said there's a gene called unicorn, which I thought was super cute too. Ben says, I've been told genetics is where the funding is. Yeah, that's a fair point. But there's other places to get funding. But genetics definitely has a lot of applications. I thought it was super interesting how Fantastic Four is another gene group from that talk. Yeah, I just thought it was really cool to think about the evolution of the parts of this seed and what leads to that, right? And how the integument changes and turns into the seed coat, and the different genes that regulate that in different members of the gymnosperms, because we don't really talk about that. I did want to jump in many times when the speaker was saying cone for things like ginkgo, but you guys have already had your exams here on the gymnosperm, so there shouldn't be any confusion. But Dr. Casper was still teaching this course, and I keep this legacy, right? Cones are in the conifers, everything else is strobili. All right, so what time we got here? 10:09. And I still don't see Dr. Georgia Silvera Siemens yet. Let me just make sure I don't have an email about a problem with connection. Claire, since you are here, would you have a problem going first if we don't have Dr. Silvera Siemens show up on time?
I can definitely go first if you need me to.
Okay, awesome, Claire. Thank you so much. So Stevens, our second speaker, is actually going to be joining us from work. So he has a very tight window that he can join us for. And so I don't want to mess that up. Okay, so if that's the case, I am going to, it's 10:10. I'm going to make Claire co-host. Yes. Alrighty, Claire, you are co-host, and I believe you should be able to share.
Okay, I don't have any slides or anything, so I can just always.
Oh, that's fine.
If you just want to show my face. So
yep.
That...
Hold on, I got a little thing here from Stevens. Okay, here we go.
That's me. Well, I can turn on my video too if folks want to see me talk.
I see you right now.
Oh, okay, cool. I can only see the presentation. So hi everybody, I'm Claire. So I worked with your professor at NYBG, which I imagine, Rebecca, you've told them about that.
Oh, actually, no, that's funny. I was going to do this whole thing. Right. So Claire and I first met, Claire
no worries.
First met when we were working as summer interns at the New York Botanical Garden in 2012. Well, it's like hard to believe it's been that long.
That's a long time ago.
He has a bachelor's in plant biology and English from the University of Vermont.
Yep.
And Claire is awesome in that she is just probably the most passionate person I know about science education and SciComm. And she's just really taken upon herself to spread the word about how cool plants are and how cool science is to a very diverse audience and age groups. She's the founder of Brilliant Botany, which is a website you should totally check out. Avid gardener and farmer, and a freelance science educator, which, yes, that is a thing, students. So you can become that. And so, yeah, thank you, Claire. Sorry, I got a little distracted because of the lineup.
No worries. I just didn't want to, if you had mentioned that you worked at NYBG Herbarium, I didn't want to reiterate that unnecessarily. So yeah, everybody, it's nice to meet you. So as Rebecca mentioned, so I do a lot of science education on the side. I'll talk about my day job, but it's not necessarily super plant related, kind of is. But when I was in college, actually, I started a blog called Brilliant Botany on Tumblr. It was the heyday of Tumblr. Which eventually kind of, it's funny, a lot of my jobs that I've gotten, I got because of Brilliant Botany, not because of like my day jobs, or a combination of both. So I started out making like blog posts. I actually have done YouTube videos. I don't have a lot right now. I haven't been making a lot, but if you go to brilliantbotany.com, that's where you can like find stuff. In non-pandemic times, I do like science festivals. I've done library programs. I do a lot of education about pollinators because I do a lot of work with kids. And one thing I really like about pollinators is one, bees and other pollinators are really cool, but also for working with kids and adults, it's a really concrete way of talking about plants and conservation because it's very difficult to talk to like a 10-year-old about deforestation in rainforests. Like it's a big problem. It's something we need to do something about, but a 10-year-old can't really do something about that. But a 10-year-old can help advocate for habitat conservation in their community or plant flowers for pollinators. So I really like it as a way of kind of engaging with people in a way that's more accessible. Plus, everybody loves bees pretty much. Even if they're a little afraid of them, they respect them as a creature. So that I really enjoy. So that I kind of do on the side. But as I mentioned, I have worked in plant science. So I worked at the New York Botanic Garden Herbarium. And then I was still in college, so I worked at the University of Vermont Herbarium, and then I worked at the Harvard University Herbaria. And in all of those jobs, I was doing digitization. So we were taking specimens, these dead, depressed plants from the collection, and taking photographs of them, and then pulling the information from the labels that go with them. What this really means is that I'm really good at reading old man handwriting, really good at reading cursive. Yeah. When I worked at Harvard, I was working on an NSF grant funded project where we were digitizing all of our New England collection. The Harvard Herbaria has, I used to know the stats better. They have like 4 million, lots of specimens, tons. I think probably in the realm of NYBG what they have, but New York Botanic Garden. But we were only doing the New England ones. So all of us on the team were really familiar with the handwriting of all of the big collectors that we had to deal with. One of the struggles of that kind of field, though, as some of you probably encountered, is grant funding is short term, and then when the grant ends, your position ends. And a lot of us would have loved to stay, but there's only so many permanent positions. I worked for a little bit on a farm doing science education. That part was great. My supervisor was terrible. Unfortunately, that's one of the things that can happen. I've worked in a couple of different museums because that kind of goes hand in hand with herbaria and like plant science as well. But then, so now I work for New England Biolabs, which is a science biotech company. We make things like restriction enzymes, which are used to chop up DNA and RNA and other reagents for research of that sort of thing. So it's not directly plant related, but it is definitely science related. I work in the quality department, so I'm one of the people that does a lot of the documentation stuff. I chase people down and tell them to update their procedures. But even though my day job isn't plants, the thing I love about plant science is that plants are everywhere. So even if you're not doing it as your day job, though it's awesome if you do it as your day job, it's something that you can kind of do on the side and still engage in your free time. So I collect carnivorous plants. That's one of them, especially during being home all the time. House plants have been like a really great hobby. You can still collect plants and press them and create your own herbarium specimens, even if it's not your day job. It's a really fun hobby, that sort of thing. And then of course I do the science education on the side when I can. So I've had kind of like a lot of people, a very meandering route in terms of my career and interests, but I still always love plant science. I don't know. I've got a terrarium behind me and there's a staghorn fern above my desk that is out of camera. So yeah, that's kind of the gist of what I do. I can chat more if anybody has questions for me.
Yeah, so I thank you so much, Claire. I'll get the ball rolling since we definitely have time for questions and students can think about what they would like to ask. So I can vouch for Claire's carnivorous plant collection. So we're friends on social media, and I did see how you mounted your staghorn fern.
Yep.
So it's super cool. Where do you find carnivorous plants? Like, where's the best
Ooh,
place
good question.
to buy those? Because I like them a lot, and I've had a hard time finding them over the years. So what tips can you give us?
That is a really good question. So I live in Massachusetts, and the way I actually got into carnivorous plants was I had a table at the New England Carnivorous Plant Show at Tower Hill Botanic Garden, which is kind of in like central Western Mass. So it's funny to me. I'm like, oh yeah, carnivorous plants. I know where to get those, but it is such a, like you're saying, a niche hobby. So if you can find local groups that have them, if it's something that you like really, if that you want to buy, californiacarnivores.com. They're one of the big, like, long time folks who have sold carnivorous plants. Though it's funny, like a lot of weird hobbies in the pandemic, I think they've gotten even more popular. So like it was one of those things before where you could go to their site and they'd have a bunch of stuff in stock. Now you kind of, they have stuff, but you kind of need to watch their socials to be like, oh, when are they restocking things?
Mhm.
But the thing I love the most, I when I would have tables at the New England Carnivorous Plant Show. At any show, even if I sell stickers and stuff to kind of fund my education work, most shows, I'm like asking people, like, ask me questions about plants if you have them. The Carnivorous Plant Show is the only show I'm like, if you have a carnivorous plant question, don't ask me, ask anybody else here, because I know a good amount, but all the other people, those are like the really devoted carnivorous plant people, and they have a lot of knowledge.
Mhm.
And then aside from that, Facebook groups is a good place to get them. If you search carnivorous plants, there's a couple Facebook groups where people do swaps and sell from their collection.
Cool. Yeah, thanks. Because it is tricky. I know when I go to a nursery or a large garden center and they happen to have them in stock, I'm always like, surprised because it's just something you don't see that frequently.
Oh, and I will give, sorry to cut you off, one
No,
quick
no.
note. If you ever decide to get a Venus flytrap from a store, which is great, they're real cool. They're supposed to go dormant in the winter. Most people don't know this, and they can do okay for like one winter where they're not allowed to go dormant. But if they survive after a little while, they'll kind of peter out, which it's not like, a random garden store is going to tell you that when you buy it. So
Okay, that's very interesting. So when you say go dormant, are we talking all the leaves, everything is not visible anymore, or?
kind of. I haven't had a ton of success like overwintering plants. I think usually everything will die back, and then like the underground root system will survive, or they'll just produce really tiny traps. Like
Okay.
you'll just find like a little bit of activity. The ways you can do it at home, a lot of the suggestions are like if you have a garage, but like a lot of young people, I live in an apartment and I don't have a garage, so you can put them in your fridge. Is one way. I keep a lot of tropical carnivorous plants because they don't require dormancy, which is because it's a little bit of a pain. So,
Right.
but
Yeah.
because they're from a tropical environment, they don't have a really cold winter, so the plants aren't adapted to need to have a period of like cold weather where they don't do a whole lot.
And I love the image of like having the plants in the fridge. Okay, we have a question here in a chat box, and I don't know if you can see the chat box, Claire.
I can, yeah.
Okay, great, because people have been like, oh my God, Tumblr. Like,
I feel like I'm aging myself too, just because making assumptions about like the age of you students. But yeah, that when Tumblr was like first the big thing, that's when I was on there. That's it. So asking about polyploid carnivorous plants, I actually don't know. That's not something I'm on. I'm like peeking over at my other computer to Google it. That's not something I've encountered, but I don't know a whole lot about it. That's a good question. I don't think there's like polyploid variants, but I'm not sure.
yeah, so just later for the recording, Ben was asking if there were some polyploid carnivorous plants at all, and if Claire had come across any polyploid variants. That's an interesting question, Ben.
Yeah.
Does anyone, you're welcome. Does any other students have questions? If not, I have more questions that I can ask Claire, but I want to give you guys a chance to pick her brain and if you have any other questions. Well, Oh yeah, so Ben says, I saw on your website a bunch of tutorials.
Yes, I've done a good number of like tutorial videos. Some of them looking back, I'm like, I probably could have done them better, but I still really like them. I've done tutorials on how to make a moss terrarium, on how to build. A couple years ago, I made a like, I got an old pallet and used like landscaping fabric to turn it into kind of a vertical planter thing, that sort of stuff. It's funny though, every once in a while, I haven't made a ton of videos. Um, but every once in a while I'll Google something, like something niche plant question that I can't remember the answer to, and one of my videos will pop up on Google, and I'm like, oh, I made a video about that. That's right. I just don't remember. But it looks like, so we have a question about pressing carnivorous plants. So whether you have to like flay the fly traps or just lay them flat, that would definitely be like your choice if you're pressing it. If you want to sit there and like very carefully open some of the traps before you press the plant, you totally can. Or you can just kind of like let it fall how it does. It really depends on how finicky you want to be. A lot of the time in for herbarium specimens, what they really want is the flowers, to be able to identify them. Because especially you lose in the actual like leaves, which in Venus flytraps is the traps, you're going to lose a lot of the color and stuff after they're pressed, which is one of the ways that they're going to be kind of identifying variants. So really what you need is the flower. So however you lay out the traps, it's more kind of artistic preference.
Yeah, and so Vivian was asking for the website, but Ben very nicely put it in the chat
Thank
box.
you.
So thank you, Ben. I guess I have kind of a question about, because you and I both worked in herbaria, and you worked at Harvard Herbaria and then the NYBG Herbaria, which are both very old school, like especially here in the USA, they're one of the very first herbaria, if not the first herbaria to exist. And so I want to pick your brain about so working in herbaria, what would you say is your favorite thing that you have done? What's your favorite part about working in an herbarium, and what's your least favorite part about working in herbarium, based on your experience?
Yeah, no, for sure.
And know that this recording will be on YouTube, so do not
No
name
worries. Oh, so I shouldn't be trash talking people?
Oh,
No,
yeah.
don't worry. My favorite part is definitely, I guess there's two things. One is the people. There's some really fun people who work in herbaria because it means you get to hang out with plant nerds.
Oh, like me.
Yeah, exactly. One of my I run a D&D game every couple weeks, and one of my players I worked with at the Harvard University Herbaria, and we just have like a lot in common. There's really great people that work in plants. And then the specimens themselves can be really, really cool. Like Rebecca, you probably remember when we were at NYBG, they have specimens that were collected by Darwin.
Specimens.
Yeah, which is, and even so, like obviously Darwin is awesome. He's not like it. What I think is almost more impressive is that when you say that to non-plant people, they understand how cool it is. Because you can say, oh, I've seen like this historical specimen that was collected in this location. It's a rare plant. That doesn't always connect for people. But if you're like, Charles Darwin collected this plant, they're like, oh, whoa, that.
Yeah.
So
we'll leave the colonialism like kind of aspect aside for the time.
Yeah. Definitely my least favorite part, and this is true kind of for it as an industry, is there's not a lot of job security, and there's not a lot of, you don't make a lot of money most of the time. Like I mentioned, when I was at Harvard, I was on an NSF grant, and that was like great for the time that I was on it. But then when the grant and the project ended, I didn't have a job. And there were a bunch of us competing for the remaining full-time position. And I'm sure this is true of a lot of herbaria. And similarly, a lot of the times herbaria aren't prioritized there's a lot of them say it's part of a university. They're not going to give a lot of funding to the herbarium because people don't see the value in it. So you can meet cool people, but it's a difficult industry to work in and stay working in unless you kind of get your foot in the door and get that full-time position.
Yep, no, that is totally true. Vivian has a question here in the chat box. What made you want to study plants, Claire?
So in high school, I was one of the kids who actually liked the plant section of my AP biology course. I still know my biology teacher from high school. And she always, talk about when she had to do the plant section of the AP biology curriculum, so many of the kids and the students didn't enjoy it. They thought it was boring. But I still remember we had big dissecting scopes, and she brought in some like cut flowers, like lilies or something, so very large flower parts. And we looked at them under the dissecting scope, and I just thought they were so cool. And I've always just thought plants are really impressive. And once you kind of think about them, really interesting. They're very different from us. I mean, they grow their entire lives, unlike a lot of other species. But it was definitely AP biology in high school. And then when I was applying for colleges, big reason I ended up going to the University of Vermont is they had a specific plant biology degree. A lot of places you can go and get a biology degree and focus on plants, but UVM in particular had a plant biology degree and a really great plant biology department. So yeah.
Yeah, I can definitely speak to the the big difference between a biology department and a plant biology department. So when I was in grad school, it was at a biology department, and it always seemed kind of like a struggle between the different taxa, right? Like you had people who studied mammals and cells and, trying to create disease or cure diseases and things like that. And then you had, people who studied birds, people who studied snakes, people who studied plants. And so it's kind of like, it felt a little like disjointed. Whereas now at my postdoc, I'm at a plant biology department, and whoa, what a difference to be surrounded by people who love plants. And it just really made a difference. So Vivian here in the chat box says, haha, pretty cool. When I studied bio in high school, I fell in love with evolution, LOL.
Awesome.
And Ben says, I'm a biology major concentrating in plant science, so I can attest to why it's not the best.
Yeah.
Yeah, it's true.
I'll say what I find funny is, so I was a plant biology major at UVM. It was a very small department. I actually don't know how many majors we had, but at graduation, so UVM, like a lot of schools, is split up into different colleges. And if you got a Bachelor of Arts, which is what I did, I was in the College of Arts and Sciences. All of the other graduating plant biology majors when I graduated got Bachelors of Science, so they were in a different college. It's just how it worked. So I was the only plant biology graduate at my ceremony. And at UVM, you carry these flags as you walk in that have your major on them. So everybody could tell, because it was physics, one person, me, holding a plant biology flag, and then directly behind me, somebody holding the psychology flag. And the person running the ceremony, when I got up to go on stage to get my degree, stood there to make sure that they didn't accidentally skip a section that only had one name in it. But props to my advisor was David Barrington. He studies ferns. He's a really awesome guy. He sat through the entire ceremony just for them to get to P to plant biology to give me my diploma, which was like, it's kind of what they're obligated to do, but like I'm sure he could have like recruited somebody. It was very sweet of him to sit through the whole thing. So
yeah, I will say that my experience working with plant people in academia, there's a lot of heart there. People who study plants, they're so passionate about it, and I feel like there's also a lot of mentorship and like a sense of like passing the torch. So it's always nice to hear stories like that, where you just felt such support within an academic environment, because that is not often the case. But I will say that people who love plants, that seems to happen more often than in other fields.
Yeah.
So that's a really great story. Okay, so we're still waiting for Stevens, and I sent Georgia an email, but does anyone else have any other questions for Claire? And if not, I will start to introduce Stevens's work. But thank you so much, Claire, for taking the time today to join us. And it's also just great to see you.
You too.
I hope you're hanging in there, during these crazy times, but I am at least happy that this crazy times allowed this to happen, because like I'm in Michigan and my students are in New Jersey and you're in Massachusetts, right? So so it's just this is like a special thing that we can do during these times, and so I'm happy we're able to do this.
Yeah, and if any of the students has any questions for me, or like especially if you have carnivorous plant questions, I can send you links to Facebook groups and things. My email is Claire, CLAIRE @brilliantbotany.com. You can reach me there, so feel free to shoot me an email.
Yeah, I'm gonna write it in the chat box. Claire, CLAIRE.
Yep, @brilliantbotany.com.
Brilliantbotany.com. Awesome.
Well, thanks for having me. I'll let you do your intro for the next person.
Yep. Thank you, Claire. All right. Let's see here. All right. So where are all my special guests? Let me let me pull up. Oh, Stevens is here. Hooray.
Sorry.
No, it's okay. Hold on. Let me make you co-host too. But I will share. Actually, here, let me pull up the slides first. Thank you so much, Stevens, for joining us.
No problem.
I'm really excited here. And so I will share this. Share screen.
Up you go.
Okay. So Stevens is joining us from work, everyone. So definitely that's.
Can you see me?
No, I can't see you, hon, but here, I'm putting up a photo of you. I don't know if you could see the photo.
Oh, yes. Oh my God, Beatrice is here. Hi. How cute.
Yeah, so let me make Stevens co-host, which is, where'd you go?
Off. Okay. Oof, my
Oh,
hair.
here. And also Stevens has to be in his car. Yeah.
Yes.
Because he is literally at work. So make Stevens co-host. All right, so let me introduce Stevens.
Am I
So
visible?
okay, so Stevens graduated from NJIT last spring during pandemic, like a freaking champion. And believe it or not, so I met Stevens in spring 2019 because I had a colleague who let me know that there was a student in her class that was interested in learning about plants and doing research with plants. And so he reached out to me and we hit it off immediately. I was like struck by his passion for all things plants from the get-go. And then he actually worked as a research assistant in my lab from spring, summer, and fall 2019. And I had many research assistants, but Stevens was by far the best. And I basically left him in charge every time that I was away out of town. And he was even a former Plant Kingdom student. So he was in this class that you guys are in right now. And so, as I said, Stevens is joining us from work. So I just really want to thank him so much for joining us today. And with that, I'll say, welcome, Stevens.
Woo.
Woo!
Am I visible? Can y'all see me?
Yeah, we can see you.
Okay, it doesn't show me my face. I have no clue what I look like. Great. Hi. So, I guess I'll start with how I really managed to graduate and get a job during a pandemic, which is like a first for, our generation. When I originally got into Rebecca's lab, I had the intentions of going for my PhD, but the more I did research, the more I realized I wasn't 100% sure what I wanted to study. And if you're not sure before going for your PhD, I mean, that's a big no. But I really did enjoy the work I did in Rebecca's lab. It definitely set me up to get where I am now. As May, or I guess it was right before the pandemic started, so March of 2019 or 2020, let me look for any biology job in Texas because it's so cheap to live there. And then the pandemic hit and all the jobs disappeared. I couldn't find anything anywhere around here until basically May. There were no options for jobs. And when May came around, a few started popping up. And I was immediately met with a bunch of no's, which was disheartening because, you expect, oh, I'm a fresh college student, I have my degree. And then you're met with a bunch of no's to jobs you really care about. And it was upsetting, but, you have to power through. And there was one company in particular called Bowery Farming, which I was obsessed with. And I finally got an interview after my third time applying, went through 5 interviews in 2 days, and they ended up saying no because they had found a candidate with a bit more experience. Oh, yeah, Bowery. It's a great company. And that same week, I got a call back from the job I'm currently at. I work at TerraSen, New Jersey. It's a medical cannabis facility. There's a picture of me cloning. When I started here, I did not even know that it was a cannabis company, which was a unique discovery when I started. um, but I immediately started working in the flower rooms, and after a month in there, I started talking to the bosses, and I wasn't happy doing flower work, and so they moved me to the vegetative growth rooms, which is what you see me doing there. I started cloning, and I immediately started excelling because I was doing work similar to what I did with in Dr. Panko's lab, which was planting seeds, transplanting, plant maintenance work, feeding, watering, things like that. And I think it goes to show that if you stick to what you know and expand your horizons a little bit, stick to what you know, stick to what you're good at, you can really excel in where you end up. Within 2 months of them putting me on the clone table for the first time, I was like unofficially named the cloning master, and they would simply just like leave me in charge of whatever was happening in the clone room, and I would just get to do whatever they needed me to do. But it was really cool to be this brand new employee to the company, you know, 3rd month here, and they're leaving me fully in charge of like the clone room, you know, oh, go into room 7 for us and clone as much as you can. You know, I come out after 2 hours with 800 clones in a tray. And I kept on with all that work, and they ended up putting me in charge of the veg room in the greenhouse, which is where I started out. Our property has a greenhouse and an indoor grow. We're the first cannabis greenhouse in New Jersey. And the indoor grow is more controlled, so you can have more fine product. And I started off in charge of the greenhouse in the greenhouse. And after a month of running that, they had me come indoors for like one day to do a clone session for them. And we got the cloning done in record time. They were saying it was probably a state record, which was like so amazing to hear. And within that month, we broke that record that we had set like 5 times. We've gotten 6,000 cuts done in 3 days, which is unheard of. And when I say cuts, I mean clones plugged into rock wool, which is our rooting medium. And the second I did that, they were like, yeah, you're not going back to the greenhouse. You're indoors now. Like, this is where you need to be. And I actually just got promoted last week, so it's really cool. I'm now the mother lead, which means I'm in charge of the vegetative. Thank you. The vegetative room full of the mother plants, which are the plants that we clone. So my job is to make sure they're maintained well, and I can get as many cuts from them as possible so that we can continue to grow and do what we need to do. But I'm also very involved in something called phenotype hunting, which is when you plant seeds and then you basically try to choose the best traits that a plant has. It's really cool. We're growing good stuff, you know? Yeah. That's a picture of one of our plants in the greenhouse. Thank you. One of our plants in the greenhouse. So the plants in the greenhouse are unbelievable to look at. They can grow to like 9 feet tall. So like when you harvest them and you chop them down, you get pelted by an enormous plant covered in resin. So like you're sticky, like you're just covered. But I never thought that I would be working with cannabis, especially in New Jersey. And I've grown to love it so much because it's such a unique plant. And I'll never forget my first day was a little rough. And my now teammate, Sarah, looked at me and she said, I want you to remember that no matter what happens here, even on your worst day, you helped a patient get their medicine. And I was like, wow, like I didn't think of it that way, but like this work is important. You know, there are people who rely on us for their medical problems. But I think it's important, general idea, to focus on what you know, focus on what you're good at, and be persistent, because if you give up when someone says no the first time, you might not get where you want to go. And you need to stay focused on what your goals are. Even if you veer off that path a little bit, you can still get back and you can do great things. I got promoted in 7 months at a company. Like, come on, anything's possible.
During a pandemic.
Yeah, during a pandemic.
Wow. So, Stevens, this is such great advice. And I want to open up, I'm sure I have students that have questions. I wanted to open this. So Brittany is asking, how do you test their quality?
So the state has set certain regulations for how we test our product. We take samples out of each batch and send them into the state, which they will test for certain things like, again, I don't know the exact parts because I'm not in the lab, but they do test for if we use pesticides we weren't allowed to use, if there's any sort of mold or pathogens present, as well as THC and CBD content. And New Jersey does not require you to test terpenes, which are like the oils in plants that smell good, but most other states do. And I'm hoping New Jersey will start doing that soon because they do have an effect on the quality of the product.
And then Brittany's asking, do you get to take anything home?
Unfortunately, no. But I mean, maybe in the future, I doubt it because the company would be losing money, but discounts. Yeah, so cannabis employees do actually get a discount at dispensaries, but since recreational sales haven't begun, I can't utilize that currently. But yeah. Sorry, I forgot that first question that I was answering. Can I access the chat? How do I do this? I haven't used Zoom in a year.
Let me see. I think it was just how do you test their quality? Which I think you answered. Here we go. Here's another question from Ben. In terms of phenotype hunting, is that controlled fertilization, or is it easier to just plant seeds and then just observe any phenotype changes? And also, what's Rockwool?
Great questions. Okay, so with the phenotype hunting, we're not involved in breeding yet. So we purchase our seeds from breeders who tell us what the 2 parent plants were, and they give us a strain name. And then when we're hunting, we plant the seeds and let them grow. And when they're in a veg room, they're getting more than 12 hours of light, which keeps them growing branches rather than flowers. And they'll start to do something called sexing. Well, they pre-sex. So they'll show little filaments at the branches, which tells us it's a female. But to be sure, we send clones into the flower rooms and they'll show us what the product will look like. And we can get a good feel for if it grows well, if it makes good product, if it smells good, looks good. And then based off of what we see in the flower rooms, we'll grow back. Yes, they are stigmas. Yes.
Just to be clear, because
Yeah.
filaments are part of the male plant, and I don't want
Yes.
people getting confused.
That's on me. Yes. I'm used to not using scientific terms here because many people don't have a background in plants. So I've had to alter my language a bit.
No worries. It's just they have a test on it, so I want to make sure there's like no confusion.
Sorry. Yeah, and then based off of what the flower rooms tell me, I'll kill off plants that are undesirable and then up the production of plants that are producing good product and clone more of it for the veg rooms. That way we can get more cuts and fill more flower rooms with it. And Rockwool is our one of the pictures in one of my slides was like a little greenish yellow cube with roots sticking out of it. Yes, that is rock wool on the right there. It's basically rocks that were melted and spun into fibers, and they can hold, I believe, 90% of their weight in water. But if you shake it out a little, it can be half water, half air, which is really good because roots need air to grow, but they also need to be kept moist. And other questions were here.
There's another question about what kind of environment, temperature, humidity do you have to grow the plants in, and does it vary depending on each kind of plant?
Great question. So we keep our plants at above 80 degrees. It does vary depending on the temperature outside and depending on which building we're in. So the greenhouse was a struggle. Some days it was 50 degrees, and I've even been in there when it was 120 degrees. It varies extremely. Whereas indoors, we have a very good control of the temperature, which helps preserve terpenes in the plants. And so typically 80, 85 degrees. We try to keep the humidity under 60%, but that's hard when you have hundreds of plants that are being watered multiple times a day because a lot of water is going into the air. And it does vary slightly with plants, but since it's a large room, we don't have much control over that. If they don't like our environment, we personally make the choice to get rid of them because it costs more to put a new room in and grow them in their own specific conditions than it would to just get rid of the strain. I see the question from Brittany that says kill them. If this is referring back to the phenotype hunting, yes, there are plants that I have to kill because, what do you mean kill the undesirable ones? Yes, so if I send a plant into the flower room and it grows branches that hit into each other, down the road, the flower buds will be on top of each other, which means there won't be airflow there. And so this will lead to issues down the road, potentially rot, which is not something we want to deal with because that just causes many issues for us and can put our customers at risk. So we kill off plants like that. There's also plants that will make very small buds, which isn't useful to us because we sell by weight. So a small bud would mean we have to grow more plants to make the same amount. And so things like that are reasons we would kill plants. Can you elaborate more on cloning? How long is the process usually? So the process for cloning is I'll cut branches off of mother plants, bring them into the propagation room and cut them to size. I have to be careful with what I say here. I don't want to share wrong information I'm not allowed to share.
Industry secrets.
Yeah, I have to cut them to a certain size, which I have marked out on cutting boards. And I put them into our hormone rooting solution, which is indole 3-butyric acid. It mimics an auxin, which promotes rooting. And the goal with plugging a plant is to how do I not say this? We want to promote healing to occur in the wound that we're causing because we are cutting a branch. But when that wound heals, it's going to form callus tissue, which is a plant's version of stem cells. And the rooting hormone on that area will form roots. Typically, we have clones in a tray for about 2 weeks before they're transplanted.
Okay, well, guys, I know that you probably could ask a million questions to Stevens, which is part of the reason why I brought him here, because I knew you guys would really enjoy hearing about his journey and seeing another way that you can make money working with plants in a different way, right? So it doesn't have to just be academia. There's plenty of different ways to study and work with plants. and do so and be able to pay bills and like be successful. So I just want to thank Steven so much for coming today. I know he's at work right now. Um, so I want to let him get back to it. But thank you so much, Stevens. I really, really appreciate it. And yeah, if any of you have additional questions that you would like to ask Stevens, you can email them to me and I can pass them to Stevens in a email.
Totally. Thank you for having me. Thanks for listening. I hope this opened you guys up to new jobs. Do well in Dr. Panko's class, please. Good luck, everyone. Bye.
Thank you, Stevens.
Thank you.
All right. Now we have one more special guest. Let me pull up here and let's go here. And then I'm going to share screen right here. And then I'm going to make Dr. Georgia Silvera Siemens a co-host. So just briefly as an introduction, here, more and co-host. Yes, and then I make this big for now. Oops, we're still sharing Steven's stuff. Boy, I got a lot of boxes up here. I gotta move things around. Here we go. Okay. So just as a little introduction, so Dr. Siemens is an alumna of Wesleyan University, the Yale School of the Environment, and the University of California at Berkeley. And basically, she is kind of like an ecological renaissance woman. she really is. I mean, her experiences include so many things, including urban and community forestry, independent research, freelance writing, eco art and mural projects, education and advocacy work. And she's also the founder of Local Nature Lab and the co-founder of Black Botanists Week. And believe it or not, we actually met on Instagram, which is not how I had thought that we would have crossed paths, because we honestly have a lot of friends and colleagues in common. So it's really interesting that that's how we met, but that is how we met. And so with that, I want to thank Georgia for joining us today, and I'm excited to hear what she has to share with us. Thank you, Georgia.
Thanks, Rebecca. Hopefully we'll meet in person in like real life, at some point.
What we have, so she and I have met together virtually just to kind of chat, but yeah, we've actually never met in person yet.
Yeah, I'm gonna start sharing. Let's see. And I've used Zoom so much this year, but I'm still a novice, so bear with me. Okay. So I'm going to talk about Washington Square Park, which is where I do a lot of my work. So the Washington Square Park Eco Projects, that initiative has been branching out beyond the park for several years. And so it's going to be a combination of birds and trees that you'll see, but of course you can't talk about birds, at least in terrestrial ecosystems, without talking about trees. So. And I should say, feel free to stop me at any point to ask questions or for clarification. Okay, so I like to talk about the fact that I am living and working on unceded indigenous land. In my case, in downtown New York City, it's Munsee Lenape land. Okay. So this is a screenshot from the Wilikia map, which was created by Dr. Eric Sanderson. And the exact date for this data, it's very strange, is September 1609. That's the actual information in the database. So you're looking at the bottom part of Manhattan, September 1609, and Washington Square Park is outlined in the orange rectangle. So it looks just pretty green, right, with no differentiation. But there's another database, New York City OASIS, and this is publicly accessible. So if folks are really interested in New York City ecology, the Wilikia map and the New York City OASIS map are two really good sources. And again, I've outlined the park, this time in gray, and the key is not actually on here, would be too small to show. But you can see that there are sort of several different things going on even within this 9.7 acre parcel. You have floodplain forest, there's oak tulip tree forest that was on the site. There was also red maple hardwood swamp. There was horticultural fields from the Lenape. So you had successional landscapes. There were beaver dams on this landscape. So sort of really rich, 1609. This is just to give you some background. And then 1871, this is what you have, lawn and trees. And the only water source being this fountain, whereas in the previous map, you could see this above ground stream flowing through the park, and it goes off the map to meet the Hudson River. In the 50s, the last car goes through the park, and this is the site today. So that's a very brief history. And what you're now seeing is the continuous line transect that we use to do our wildlife surveys. And we're typically just seeing birds. So other non-bird wildlife we see are mice, rats, squirrels. We have seen a raccoon. We do take note of insect life, but my colleague and I are not insect specialists. So we sort of use iNaturalist for that. We do have a pretty good insect population in the park, but of course it could be better if more native trees and shrubs are planted. So we see a range of butterflies, native bees, and native flies as well.
Georgia, I don't know if you can see the chat box, but there's a question from Brittany just for clarification. If that first aerial pic was from the year 1609?
It was, and so it was, there wasn't aerial technology in 1609. So it's actually the scientist's version of what the landscape probably looks like in 1609. So it's all computer generated. It's a computer generated aerial and not a true aerial of 1609. So our transect is 6.65 miles. And this is just some summary data to give you a sense of bird life in the park. So we started conducting the survey in 2016. We typically do it bi-weekly. You can see that itself also varies. Last year was actually a pretty good year in terms of the number of unique bird species we saw over the course of the year. Of course, last year was the height of the pandemic, and so spring and fall were quieter than usual in terms of human-generated noise. And even though we don't count herd-only birds, we did hear more birds than we usually did, so then we went to look for them. Washington Square Park is an urban site. So when you're thinking about urban ecology, of course, it's on a gradient, and Washington Square Park is not Prospect Park, for example. It's 9.75 acres in a highly residential neighborhood in the city. And so you're gonna get the complement of the most populous bird species. So house sparrow, pigeon, and starling, definitely always in the top 5. But you do see American robin and white-throated sparrow in the mix as well as mourning dove. This just shows you, kind of breaks down the canopy layers in the park. And I've also included some of the plants you'll find in each of these layers, as well as some of the birds we've cited in each of the layers. And this is useful for us just so when we do advocacy work with the Parks Department or elected officials, as well as something that we use when we do our educational work in the park. And to sort of then hone further in, so the park is already small at 9.75 acres, but through our survey work, we've identified micro hotspots in this small hotspot of a park. And I'll just sort of walk you through them. So in the northwest corner, where we definitely get that sort of 5 layers of forest, we see, and we also have a lot of oak trees in that section of the park. We see a lot of warblers in that area. So spring and fall, you go into the northwest corner and you will see a diversity of warbler species. So that's always really exciting. The red arrow indicates one of the crabapple groves in the park. And I'm not quite, for me, it's really hard to tell crabapple to the species. So I always just say crabapple to the genus. I don't know if these particular crabapples are native crabapples. And of course, we could have a conversation about native versus introduced species in the city, but these trees are just abundant with fruit in the fall and hopping with birds. And so you get thrush species coming through. So first you get robins, you get hermit thrush, you also get catbird, you get cedar waxwings. All the birds that are loading up for fruit in this season flock to this group of crabapples. Moving east, this section of the park, there are spruce, I think it's red spruce. There are also deodar cedars. And again, this is a nice little area to look for birds that like to hang out in coniferous forests. So we do see a lot of kinglets in this area, for example. And because the ground is quite dark and shady, because it's a bit of a coniferous zone, you do find this sort of secretive thrushes in this area as well. So we really see a direct connection between the types of plants and forest structure in the park and the types of birds that use the park. So going west again, this purple arrow is pointing to one tree, and it's the northern catalpa. There are 2 in the park, but this tree is often where we see a lot of American redstarts, which are, which like a lot of other birds like to feed on insects. And for some particular reason, the catalpa attracts a lot of insects. The orange points to quite a big lawn, and there are other lawn areas in the park where we see a lot of sparrow activity. And it's not just the house sparrow, which is a disparaged bird, but we see migrating sparrows as well. So the white-throated sparrows, which are very common, they overwinter. We also see chipping sparrow, song sparrow, fox sparrow. It's just a really great place to look for sparrows and other ground feeding birds. And then finally, this is not in the park and it's not even tree related, but this building belongs to NYU. And there's been a nesting pair of red-tailed hawks since 2011. And I'll give more detail about the hawks later and how they use the park. Okay. So just to spotlight birds, just to let you know that, the plants really make the park. So in 2017, we had the good fortune of hosting the Kentucky warbler in the left. And Kentucky warbler is not really a bird that you would typically find in a small urban park. But that season in particular, the understory sort of ground cover was really thick and lush and much more contiguous than it is now. And so this was just a really prime location for this bird to spend several days during the spring migration. So it stopped over. So it wasn't kind of a accident. It was here for an hour and left. It spent a fair amount of time in the park. And then another surprise but welcome visitor, black-crowned night heron. This bird also, Washington Square Park is not its optimal habitat, but it was here for several days. And we sort of joke that maybe it has marsh memory, somehow this sort of generational memory that this landscape used to be ideal for herons. Another tree bird association, for those of you who are not bird watchers, this is the yellow-bellied sapsucker. It's actually my favorite woodpecker, if not my favorite bird. And this is one of the hawthorns in the park. And you can see that this sapsucker is not necessarily the sapsucker that made these holes. This is also an image on the right from a tree in the park. But you can see the bands of sap wells that the sapsucker makes. And they usually start by making a horizontal band. And if the sap is running, then they do vertical lines. The sapsucker is really important because not only is it mining for its own food, but the wells, when they run, attract insects, which the sapsucker also eats, but you also get other birds coming to sap wells like hummingbirds. So the sapsucker is sometimes referred to as a keystone species because of the resources that it provides to other animals. And here we have the hawks. So their nest is in this window penthouse, and the nest was first built in 2011 and is renovated each year. This year it's a brand new pair. This is a photo, I think, from 2016 that I took off the hawk cam. You can see the birds come into the park and they use the trees in the park, both in their courting behavior and also to repair the nest. They also use the trees as perches from which they hunt, and then they also bring back the food to eat on tree branches. You can see that the male on the right has food in his talons. This is this year's pair, and the female can be identified by the whites above and under her eyes. Okay, and this is just here just to show you that the pair was pretty consistent, the male, for several years. And the female actually, unfortunately, changed a lot over time due to unfortunate circumstances. And this year, as I mentioned before, it's a brand new male and a brand new female. And they are unnamed, thankfully. So going back to trees, this is one of the first projects we did, which is an online tree map of the park. And there are approximately 340 trees in the park right now. Representing 32 species, 33 if you consider the holly a tree. There are actually two different types of holly in the park. One is a tree form and one is a fastigiate shrub form, but that might be getting into the weeds. So one of the projects we have specific to trees is a phenology monitoring project, and we use the Nature's Notebook app that's part of the National Phenology Network. And our goal here is a couple of things. So the park doesn't have its own phenology database, and phenology means the study of seasonal changes in plant and animal organisms. So if you think about, you're probably really familiar with phenology, I'm sure, from Rebecca, but just in your own lives, things that you take for granted, buds opening up in the spring to show leaf or flowers, darker green leaves in the summer is one phase. Then in the fall, you get fall color, you get leaf fall. And then with birds and other animals, the sort of stages they go through. So finding a mate, making a nest, hatching eggs, fledging, that's all part of phenology. And scientists use this data for different sorts of things, including to talk about the impacts of climate change on ecosystems. So in the park, we're monitoring the trees indicated on the map, and I'll sort of go through them with you. This is a flowering dogwood. We have two Kwanzaa cherries, one here and one here. I hope you can see my mouse. This is a star magnolia. We have a silver and red maple in the northwest corner. This is a tulip tree, a sweet gum, which we added this year, in part because we have heard that in New York forests, at least, sweet gums are becoming co-dominant with red oaks in the canopy. And so we thought it might be nice to study that tree since we have it in the park. We haven't yet added a northern red oak to our study. And that's really kind of for logistical reasons. So there's a really lovely red oak, but the canopy is quite high, and that does make it difficult to do observations. There are younger red oaks in the park, but they're in the playground. And legally, you're not supposed to enter the playground unless you're accompanying a minor. So something we have to work around. So sweet gum and a Yoshino cherry. And I forgot to mention, this is a Kusa dogwood. And so we started this project in the fall of 2019 with some really wonderful volunteers, including an ecology methods class at NYU. So we record things like swelling flower buds. So this is the this year's growth of a Yoshino cherry. Open flowers, this is a star magnolia. You can see that it's exhibiting leaves as well. This is the flowering dogwood. We're actually looking at 2 dogwoods in the park, the Kusa, which is an introduced species, and the flowering dogwood, which is a native species. And these white structures are not the petals, they are bracts, very showy. The flowers are actually in the center of this brilliance, and you can see 2 phases of flowering here. These flower buds aren't yet open, they are swelling, and this is an open flower bud. You can see the reproductive organs. We definitely look at newly emerging leaves, changes in leaf size. This is a tulip tree, and you can see that its flower bud is swelling. This is some early data that we have starting in 2019, just to give you a sense of what the data looks like visually. This is known as a phenophase calendar. So we're looking at the leaf phenophase and the colored leaves phenophase, and there is some overlap. We're looking at the species red maple for the years 2019 through 2021. Pardon me, my cats would like to leave. And the red lines indicate that the observer was in the field and observed this phenophase, and the gray lines indicate that the observer was in the field and did not notice the phenophase. So you can also see there's sort of consistent observer activity, and then you can see where there are gaps in observation. For the red maple, you can see how long once it's in leaf, how long the leaves are on the tree, and when fall color is first observed and when it is observed for the last time. So our goal is to sort of do some version of a study like this, where we can talk about the impacts of being in an urban setting, being along an urban heat island gradient, and how that might impact leaf out, the timing of leaf out, and the timing of flowering. This particular research, the researchers found that there is an impact, though it depends on where you are in the country. It turns out that colder areas are actually experiencing earlier leaf out and flowering. Colder areas being impacted by the urban heat island effect are experiencing earlier leaf out and flowering. It's not necessarily the case in warmer areas where plants might be less sensitive or the urban heat island effect might be dulled by the fact that you're already in a warmer climate. Oh, I see things in the chat. Oh, okay.
Yeah, we're just, I'm pointing out, like they're doing a phonology assignment, so I'm pointing out to them, like, I'm not the only person who is running around with this word phonology. There's a whole bunch of us that are interested in phonology, and then also that we did cover Brax in a previous lecture as well. So I was making the point to say, Brax, guys.
Yeah.
So just so you know, probably have just a few more minutes here, and I also want to have time for questions.
Okay.
So I just want to let you know.
This is the last of 2 slides, I think. Or there are 2 more slides. So this part of, so going back, urban heat island effect impacts on timing of particular phenophases. We are starting to think about a phonology research project that looks at several sites in New York City that are also collecting phonology data. So using an urban rural gradient with the Ward Pound Ridge Reservation, which is outside of the city proper in Westchester. It's the same hardiness zone, but definitely off the sort of urban heat island radius. And so these are the 5 sites in the city. We've got New York Botanical Garden in the Bronx, the Ramble, which is a natural area in Central Park, Washington Square Park, Greenwood Cemetery, and potentially the Brooklyn Tech High School site. They're looking at a park in Brooklyn. So Washington Square Park again. And this is Greenwood Cemetery. This is where their tulip tree is and where their flowering dogwood is, which we are gonna see in the next slide as a comparison. So looking at Washington Square Park and Greenwood Cemetery, we're looking at the flowering dogwood and just looking at one phenophase over time, leaves or needles. And again, the colored lines mean the phenophase was observed in the field, and the gray lines indicate that that phenophase was not observed. It's very limited data. It's only 2 years. We would need at least, I would say, 5 to 7 years, if not 7 to 10 years. So this is definitely a long-term study. Again, you can see there's some differences in observation consistency. This is definitely probably at least in 2020 because of the pandemic. In Washington Square Park, we've been more consistent with our observations. So this was April 5th, so we definitely have more data here. And you can just see, comparing the 2 sites, perhaps, their leaves stay on a bit longer in Washington Square Park than they do in Greenwood Hills Cemetery. And we can ask why that can be when we have more data. And that indeed is my final slide. Thank you.
Yay, thank you so much, Georgia, for telling us all about that today. I really love the overlap of, again, the phonology assignment, because they do a 6-week observation period of about 10 trees in their neighborhood. And it's just nice. I've been doing it for 4 years. I've told students the goal is to hopefully use this data set, because I do it every year for a little mini study, but it really depends on the quality of the pictures I get, if I can actually tell what species we're looking at.
Yeah.
Really great presentation. I want to open up the floor to any students who have questions for Georgia. And I guess while you guys think about that, I'll ask a question about, the importance of learning about plants, but also exposing people in urban areas to plants. So I know you're so passionate about this. What do you think? What drives you? What do you want people to know in urban areas about plants? Because you work with so many different age groups and
yeah. So
It's a big question, right?
No. So a couple things. So I grew up surrounded by trees. They were fruit trees, but so I think my early exposure sort of primed me to see trees, to notice them. And I am not of the school that thinks people are not having experiences with nature in the city. I actually get a little bit frustrated when I hear that narrative. But there are many more opportunities for people to be aware of plants. And it's not something that's emphasized in school. It's not something that's emphasized in pop culture. Even within the two worlds that I straddle of bird watching and trees, there definitely, birding has much more cachet in popular culture than tree watching. And I think that's something that plant folks secretly want to change, but we haven't figured out the best strategy. I mean, plants in cities do so much and they do it quietly. And I think that's partly why they're overlooked, right? We pay attention to them when they're in flower, and then we totally forget about them until it's fall. And so part of this sort of phenology monitoring and recruiting volunteers to do it, because it is an year-round project, is to use the showier phases of trees to bring people in. Because even when trees aren't being splashy, there are all of these minute changes that are so fascinating. And you only notice when you're standing in front of your tree and you're being asked to pay attention. And I would like people to be as familiar with trees as they are with, I don't know, like the latest person on TikTok or whatever, or the latest show on, I don't even know, Hulu or something.
Same. I also want that. Oh, here we have, hold on, I got some things in the chat box about hawks. Do they nest on the lower parts of trees or no?
So the hawks that are associated with Washington Square Park have never nested in a tree in the park. They've only used that building. There is a park that's a little bit larger than Washington Square Park in the East Village. It's called Tompkins Square Park. They also have a nesting pair of hawks, and those hawks use trees in that park. And they've actually used two different trees in the park, but they're never low, they're never sighted low. They're always sighted towards the top of the tree. And I think that's for a couple of reasons, well, a few reasons. One, just architecturally speaking, there's just gonna be, they choose a site that is sort of strong enough because it's quite a large nest. You sort of don't get a sense of that unless you're right on the site, how large the nest is. They also need to have like 360 visual access. And I mean, the hawks don't really have predators in the city, I guess, unless you're talking about egg predation.
Or
But
trucks?
yeah, but hawks like to be up high,
Yeah.
and so they tend to site their nests and their perches up high.
And then there's another comment in the chat box from Ben about, I don't know if you've met Dr. Hulsefeld at Rutgers, but Dr. Panko should introduce you to. He's all about plants and birds, and you two would be birds of a feather.
I like that pun. I will write down.
You know Klaus?
No.
Oh, I could totally introduce you. Yeah,
Okay.
he does a lot of work in Liberty State Park in Newark. That's kind of been his baby, as it were, because it's just been a long time that he's been looking at the different composition of plants there and birds. And also Liberty State Park, I believe, used to be a Superfund site. And so the chemistry of the soil has changed a lot over time, but obviously still has a lot of issues. So but I could totally introduce you guys. So
Great.
Yeah.
Thanks. And I do want to say, based on one of the comments that the last presenter made, that I would encourage you all to consider a career in environmental education at whatever level. It could be, obviously at the university level, one could be a professor. But I think as more and more people live in cities, and as more and more people need to reap the benefits that nature in cities can provide, it becomes really important to build people's awareness and knowledge about the environment in which they live. And so education is one way that that can happen. And I can't see any of your faces, so I think it's also really important for urban educators to represent the diversity of the communities that are living in cities. And so, yeah, just a plug for environmental education at whatever level interests you.
I think that is a great place for us to end for the day, because I know students have to move on to the next class, but what a great message. Yes, we need representation, and we need knowledge of plants and other aspects of the ecosystem. You know, if you have a young child, right, you've been doing this phenology assignment, you can show the young child the changes that have occurred over time just on a walk. You don't have to end up, you know, becoming in the field and working in an academic institute to pass on your knowledge of your environment to those around you. So I think that's a really strong message and thing to keep in mind as you guys move beyond this class, because we have one more meeting on Thursday where we have some more special guests. And then after that, you guys will be on your way to your next journey. So thank you so much, Georgia, for meeting with us today. It was just a great talk. And thank you, students. And I will see everyone saying, thank you. Yeah, thank you so much. And yeah, I'll see you guys on Thursday. Thanks again, Georgia.
Thanks, Rebecca. Thanks for your patience. Bye.
Bye.